Okay, so let's finish off section 3 by talking about viral replication. So remember, we can have several different genome types when we're talking about viruses. They could be DNA or they could be RNA, and they could be single-stranded or double-stranded or segmented. So we're going to start off with our DNA genotypes. So when DNA viruses infect animal cells, again, they're typically going to rely on the host replication machinery, but their strategy in which they go about replication is going to be different depending on whether or not their genome is going to be double-stranded or single-stranded. So for double-stranded DNA viruses, the replication process is going to be very similar to the host cell DNA replication because it's using their machinery. So after the nucleic acid enters into the nucleus, the viral DNA is going to be transcribed, again using the host RNA polymerase. It's going to produce the mRNA that's then going to travel to the cytoplasm to where the ribosomes are located, be translated into viral proteins. And then these viral proteins and enzymes, like things like the capsid proteins, they're going to have to be transported back to the nucleus. And that's where the viral genomes are going to be assembled and eventually turned into new virions. So not all double-stranded DNA viruses act in this manner. There are always some kind of unique replication strategies. So one example is hepatitis V virus. So even though they're classified as DNA viruses, they actually use reverse transcription. So they create an RNA intermediate and then convert it back into DNA. So this actually makes them more persistent in host cells and kind of complicates immune clearance and trying to get rid of them. So again, viruses don't always follow these exact same steps of their typical genome type. They have kind of evolved these creative mechanisms to help maximize their replication. So this ability to kind of repurpose cellular machinery is what makes many of these viral infections very difficult to control. So single-stranded DNA viruses or ssDNA viruses, a good example is parvovirus B19. This is really challenging because host cells don't normally replicate single-stranded DNA. So in order to solve this kind of challenge, the viral DNA actually folds into this hairpin-like structure, and that's going to provide the starting point for DNA polymerase. So the host enzymes then are going to synthesize a complementary strand, forming a double-stranded intermediate. From there, the replication and transcription is going to proceed just like we talked about. So this is a really interesting strategy. It's common in very small viruses with very small genomes. Because they lack many of their own enzymes, they're really completely dependent on the host cell in order for their replication cycle to proceed. So they normally are going to infect rapidly dividing cells, so cells like those found in your bone marrow or bone marrow precursors, because those have the most active DNA replication machinery.